Hello, cell biology fans. I'm sitting here in my office, and what I'm going to do today is give you this flipped classroom lecture. I'm going to lecture about the chemistry of cell biology that you'll need to know. This is absolutely something that will be on the exam, and you also need to have watched this prior to our second meeting, Wednesday the 28th, I believe, of August. So let's just get started. As most of you know, chemistry and biology are intimately related. The chemistry of atoms and molecules really are the building blocks of everything in biology. Biology is the study of life, and life is made up of chemicals. We're going to talk about what exactly the elements are that are the building blocks, what those elements form, and what the building blocks of biology are, and how this is all connected. As you're going to recognize throughout the course, I believe firmly that understanding the structure and how that leads to function can get you pretty far in the biological fields. We function here on Earth because of our chemistry. The chemistry is what allowed us to evolve into who we are, and you're really going to see that changes in structure, even little changes in structure, can have dramatic effects on function, particularly proteins that are important for your health and well-being. Science really is divided into sort of little sections, and science, to me, I'm in this very little group of things that I'm interested in, and I'm really not a chemist, okay? But I recognize the fact that chemistry and understanding how atoms interact with one another, which is the field of chemistry, and then the field just beyond that, right, subatomic particles, that's physics. And so physics is probably the basis for all of life. The physics of it, and that leads to the chemistry. And if you look at this chart, that leads to the formation of building blocks such as amino acids, carbon-containing molecules such as sugars, nucleotides, lipids, everything you can imagine. And as we go up the scale, right, we get further and further away from the building blocks, but it really is the building blocks that lead to the formation of cells, tissues, humans, other organisms, and ultimately to cultures and civilizations in the world, right? Who studies cultures and civilizations? That's anthropology and sociology. Those are pseudosciences, so, that's what we call them. And as you go down the scale, right, different kinds of scientists study different fields in here. And I'm a cell biologist, so I stick to tissues are really a little bit out of my realm. Cells I'm really good with, and understanding maybe the next two levels down is important. So probably the most important thing for you to understand is water. Water is what allowed us to evolve into carbon-containing molecules that it's interesting, water doesn't have a carbon in it, but water is what drives the formation of life. And what is it about water that you need to know? If you know the structure of water, right, it's an oxygen that has two covalent bonds to hydrogens. What is it about oxygen that's really important? Anybody know? Anybody out there? What's the number one thing about oxygen? It's highly electronegative. That means, all right, this is the way I think of this, oxygen is a little bit of a pig, and it sucks electrons closer to it than in a covalent bond, than closer to the other atom that it's attached to. So in this case, when hydrogen is attached to an oxygen, the shared electron between the two is closer to the oxygen, creating a slightly negative charge on the oxygen and a slightly positive charge on the hydrogen. And that slight negative and slight positive charges is really what drives most of the chemistry of biology. Okay, if you look at this chart, you'll see 70% of the body is made of water. 90% of your lungs are water. 91% of your lungs water and you've drowned. Probably 89% of your lungs water and you've probably died. Blood is 83% water. Without water, we wouldn't exist. if you look, right, 30% of the chemicals, polysaccharides, proteins, RNA, DNA, phospholipids, molecules, ions, blah, 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 which one's most important? Proteins. So we're going to talk a lot about the interaction of proteins, polysaccharides, nucleotides, phospholipids with water and how that makes you become a human being. If you look at just elements, right, the main elements that make up human beings, hydrogen, carbon, oxygen, and nitrogen. And the beauty of that is if you understand the chemistry of those 4 atoms. The chemistry of 4 atoms can get you through this class. Hydrogen, carbon, oxygen, nitrogen. What's not alive? Right? The things that are inanimate, the earth, what else, what other molecules do they have? You see, oxygen is the number 1 element that makes up the Earth's crust. Calcium, sodium, magnesium, potassium, aluminum, other molecules, right, are important for non-living beings. But really, understanding the 4 atoms, hydrogen, carbon, oxygen, nitrogen, will get you really far in this class. What do you need to know? Well, everything, all of biology, really has to do with the interaction of atom to atom, molecule to molecule. I hope from 107 that some of you remember what an atom is made out of, okay? An atom has a nucleus, okay? That nucleus is completely different than the nucleus of a cell, all right? So what's in the nucleus of an atom? It has protons and neutrons, all right? Both of those have weight, all right? They actually have some mass to them. What surrounds the nucleus is the cloud of electrons. Electrons don't have very much mass. We basically consider them to have no mass. So if you look at this carbon atom, right, a carbon atom has an atomic number of 6. So the atomic number, I don't even care if you know about atomic number, but what really matters here is if you look at this carbon atom, it has 6 protons. How many neutrons? 6. It actually has a molecular weight of approximately 12 Daltons. How many electrons does it have? 6. So carbon is not charged because it has the same number of protons and neutrons. Now, if you look at a hydrogen atom, much smaller atom, it has 1 proton in the nucleus. There's no neutrons. It has a molecular weight of 1 Dalton. It has 1 electron, also not charged. So when you start manipulating these atoms, if you take away the 1 electron from hydrogen, right, it now has a charge of +1. It's no longer called hydrogen. What is it called? It's called a proton, right? Or what else? H+. All right, so I hope you're following so far. This is just chemistry, things that you should all know. The nice thing about the 4 elements that make up living beings, hydrogen, carbon, oxygen, and nitrogen, is that in order to fill their outer electron shells, what you're going to see is that one of them requires 1 electron, one requires 2 electrons, one requires 3 electrons, and one requires 4 electrons. And that is the basis of covalent bonding, filling the outer electron orbitals, right? Hydrogen has 1 electron in its outer shell. Its outer shell, right, is the innermost shell, which only can contain 2 electrons. So now, right, 2 electrons fills that outer shell. So it only needs 1 to complete itself. Carbon, right, carbon, the basic forms of life, we are carbon-based life forms, most of us, some of you may be aliens, but it has 4 electrons in its outer shell. Its outer shell can contain a total of 8, so it needs 4 electrons to fill its outer shell. The filling of the outer shell completes the molecule, and in this case that means that carbon can form 4 covalent bonds with other molecules, other atoms, or, right, it can do double bonding or triple bonding, etc. Nitrogen, how many does it have in its outer shell? Anybody? 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 So it needs 3 to complete itself, which means that it has to have, how many? 5 in its outer shell, so 3 to complete it. And Oxygen, right? Oxygen needs 2. So that means it has 6 electrons in its outer shell. If you don't remember this, please go back and read your introductory 107 materials. Covalent bonding, we all should know what covalent bonding is. Covalent bonding is the sharing of electrons. That's a really strong bond, okay? But biology is going to be really a combination of not just covalent bonds, but all the non-covalent interactions that allow one molecule to interact with another molecule. So you need to know what the non-covalent interactions can be. So there's ionic bonding, hydrogen bonding, van der Waals forces, and hydrophobic force, and we'll discuss each of those separately. I love this picture. This is a picture of a cell, the outer edges of the cell, 3D wise, and you can see, you know, there's a cell in there, the little light green, but all of these things are sticking out of the cell. Some of them are covalently bound, some of them are transmembrane proteins, some of them are non-covalent, non-integral membrane proteins. So what you're going to see is that we're going to talk about how this cell can form through the formation of covalent and non-covalent bonding. As I was saying, since this is biology, what we're really interested in is taking these elements and making molecules, right? So the molecules that get made can be on this top of this image: sugars, amino acids, nucleotides, phospholipids. That's made through covalent bonding. But after covalent bonding occurs, we have the formation of what we call macromolecules, globular proteins, RNA molecules. Non-covalent bonding creates the actual structures that guide life. All right, so we're going to talk about how proteins get organized, how nucleotides are made, how nucleotides interact with proteins, and you're going to see a lot of this has to do with the non-covalent interactions. So let's just talk about covalent for a moment. This is one of the simplest cases. If you have 2 hydrogen atoms, right, they come together, they share their outer electrons, which now each hydrogen atom has 2 electrons in its outer shell. It feels completely good, right? If you have 2 atoms and you bring them too close together, the nuclei don't want to be that close together and they will repel one another. And that's due to van der Waals forces, all right? If they're too far apart, they can't attract one another. And so there is a distance that's optimal for that. And I often talk about this, you know, if you're talking to somebody and you get really close in their face, right, you're in their personal space, there's some optimal distance for these kinds of interactions, just like there is with atoms. So this creates hydrogen, right, the gas hydrogen. As I was saying earlier about water, the beauty of this is that some covalent interactions are non-polar, which means they don't have any charge associated with them. But if you take a look here at water, the electrons are pulled closer to the oxygen atom, creating that slightly negative charge on the oxygen atom and creating a slightly positive charge on the hydrogens associated in that covalent bond. And that's, you're going to see polar bonding. So polar means that they have opposites. So think of polar opposites. So, right, you have negative and positive charges. You're going to see there's other uses for the word polar, and we'll discuss that later. Think of polar as just being opposites. Okay, in the bottom, right, oxygen, non-polar. It's sharing the electrons equally because both oxygen is pulling, and it's an equal interaction. Non-covalent bonds, they're way less attractive, and I mean attractive in they don't have a lot of power or strength. So these non-covalent interactions can come and go way more quickly than a covalent interaction. Covalent interaction is very strong and difficult to break. So non-covalent interactions are transient. Some can be stronger than others, but it really is that you have strength in numbers. When you have multiple non-covalent interactions, you may have thousands of non-covalent interactions, and those interactions add up. At any individual time, if you have 1,000 interactions, right, 500 may be on, 500 may be off. even if you have 600 off and 400 on, things are still going to be bound to one another. So think of strength in numbers. Ionic bonds, right? I hope you all remember what ionic bonds are. Ionic bonds are these electrostatic, which means there's going to be charges involved. And it's completing the outer shells of atoms. And what that does is if one atom gives up an electron and the other atom receives an electron, right? If you receive an electron, you have now a little bit more negative, so you are now a negative ion. If you give up an electron, you are now a positive ion. The classic example for this is sodium chloride. Sodium chloride, the sodium atom has 1 electron in its outer shell. It would need 7 electrons to fill it, and that doesn't happen. It's way more likely to give up 1 electron, and it gives it up, in this case, to chloride. And the chloride ion accepts that electron because it has 7 electrons in its outer shell. It wants 1 to fill it. So now you have a chloride ion that is fulfilled. You have a sodium ion that's fulfilled. Its outer shells are full. And there is an attraction between those two ions, right? The positive and the negative charges attract one another. There is energy stored in that attraction, just as there's energy stored in a covalent bond, right? Every covalent bond stores quite a lot of energy, and breaking that bond releases that energy. Here we have the same thing. There's a bond, it's called an ionic bond, it stores energy, and it stores less energy than a covalent bond, but it still stores energy. So the classic example is always sodium chloride. I will play that sodium chloride love song for you in just a little bit, but I'll come back to that. So just be clear, covalent bonds, sharing. Ionic bonds is a transfer, all right? The electron is transferred from one molecule to another, and that creates ions. Van der Waals is the really interesting interaction, and it's all about what is the appropriate distance to be separated from one another. And if we're an atom, right, there's that repulsion if you get too close, and if you're too far away, you cannot possibly attract one another. I think this is true of people as well, right? And so if you look at this graph, attraction versus repulsion on the y-axis, and the distance between the nuclei on the x-axis, so the further apart two nuclei are, you get neither attraction or repulsion. It just goes away off to the right. There's an optimal distance, right? The attraction is optimal at some distance between the two atoms shown with the line, and any closer than that, you start losing that attraction, and at some point it's not an attraction anymore, it's a repulsion. Two atoms too close together, they just want nothing to do with each other. Okay? So van der Waals is pretty easy to understand. It has to do with too close, too far away. Hydrophobic force I also think is pretty easy to understand. Okay, if something is hydrophobic, right, it means it doesn't want to be anywhere near water. And this picture showing you oil and water, or maybe, I think it's oil and water, okay, oil and water, they're still touching one another, right? There's no way to separate that in a glass. But what the oil can do in this case, and the oil is lighter as far as molecular weight goes than water, what it does is it minimizes its interaction with water. You can't get away from this interaction, but it's minimized it by putting all of the oil up above. It's not mixed, it's not interspersed. So in this case, right, we're talking about minimizing. So hydrophobic force, things that are hydrophobic want to stay together, things that are hydrophilic want to stay together. And when we have polar or non-polar molecules, right, polar usually means there's a charge. Polar molecules love water. Non-polar molecules don't love water. They want to actually get away from water as quickly as possible. So just know the difference between hydrophilic and hydrophobic. What is amphiphilic? What is amphipathic? So what is ambidextrous? Right? It's not quite the same, but it's on the same, right, same lines. Ambidextrous means, right, you can use either hand to write, to do whatever. Amphiphilic means that the molecule has both a hydrophilic and a hydrophobic portion. Amphipathic, same thing. Hydropathic means loving, right? So I love water, I hate water. So we have molecules, right, that can go either way. And they do. And what happens is they partition the part that's amphiphilic, loving, right, loves water, will go towards the water side of a reaction, and the amphiphobic, right, would go towards the non-polar portion of whatever that reaction is. We'll talk more in detail about this in a minute. So let's focus on the molecules that make up biology, right? We've said what the main atoms are, right? And hydrogen, oxygen, nitrogen, carbon. I always forget carbon. I could hand you a glass filled with these 4 atoms, and I could say to you, make a person, right? Could you make a person? Well, probably not, okay? What would be required? A lot of energy is required. Okay? What else could you make out of that glass that I gave you with hydrogen, oxygen, nitrogen, carbon? Could you make a bacteria? Yep. Could you make something in the class of archaea? Absolutely. We're made out of the same exact molecules, okay? Same exact atoms. Those atoms get made into amino acids, lipids, sugars, and nucleotides. And you need to know how these work. So we're going to talk about these 4 families of carbon-containing compounds: sugars, fatty acids, amino acids, and nucleotides. You should be able to recognize the structure of a carbohydrate, of a sugar, right? CH2O to the nth. CH2O, 1 carbon. Okay, if you have 2 carbons, it's C2H4O2. Okay. Most carbon sugars that we think about have 5 carbons or 6 carbons, right? A pentose or a hexose. So the upper image here shows 2 images of which one? Hexose. There's 6 carbons, right? I don't care, right, if you recognize a chair conformation on the right or just the structure that's shown on the left. But if you see this kind of structure, right, what is it really made out of? Carbons, hydrogens, oxygens. Nothing more. So what does a sugar do for you? It's a ton of covalent bonds that are storing energy. We can break those bonds and harness the energy for our own utilization. Okay? If you have 2 hexose molecules that are bound to one another, they're bound by, in this case, it's a, what is it called? Does anybody remember? It's an alpha or a beta 1,4 glycosidic linkage. Right. Beta 1,4 glycosidic linkage, we don't have an enzyme that can break those down. Those are the carbohydrates that are part of chitin or cellulose. All right. But glycogen and polysaccharides, those have an alpha 1,4 glycosidic linkage, which we can easily break down into monomers of sugars that we can then use to make energy. So this image, like I said, I don't care if this is alpha or beta, up or down, I don't care, but you should be able to recognize this as a molecule of sugar. That's a disaccharide in this case. It has a 1,4 glycosidic linkage, and it can be used to store energy. Here's a perfect example of energy that's being used for another purpose. Okay, these are sugars that are used for mechanical support. Chitin and cellulose are both very rigid. On the picture on the right, that is something called a potato bug. It's in the family of locust, or what's the other bug that I'm thinking of? Oh, it'll come to me, right? These guys are nasty, ugly. I have them in my yard. They live on tree roots. And the bottom left is obviously a cockroach. And right when you step on these when you step on these bugs, what do you hear? You hear a crunch. That crunch is the hard outer covering of the exoskeleton, which is made of sugars, made of chitin. Chitin happens to be one of the most abundant molecules in the whole planet. Okay, cellulose, right, is crunchy, right? You eat a piece of celery, you hear that crunch. That's because you're biting through cellulose. It's actually hard to break these bonds. So there's a lot of energy stored in there, but they also provide mechanical support. So sugars have multiple functions. Fatty acids, the second one of these 4 carbon-containing compounds that are important for the structure and function of cells. Okay. Fatty acids, if you look at these, all right, what do you see? You see a chain of carbons, all right? In the case on A, you have a hydrophilic carboxylic acid head. So that's pretty common for this to have a carboxylic head, and notice there's a negative charge, so that negative charge on the one oxygen plus the other oxygen that has a double bond to the carbon, that's going to have a slightly negative charge on it as well, because oxygen is highly electronegative and sucks the electrons from the carbon closer to it. Those negative charges, right, are going to want to interact with water. But as you go down into the fatty acid, it's just fully hydrocarbon molecules. Each carbon is completely bonded to 4 other molecules, 2 hydrogens and the 2 carbons on either side of it. So those carbons don't want to have anything to do with water. They're full. Their outer shells are full. So they're actually hydrophobic. So these, right, all of these pictures right here show molecules that are amphiphilic. They have one side that wants to interact with water, and they have one side that doesn't want to interact with water. The hydrocarbon tails can change a little bit based on size and based on whether they have single or double bonds. So what do you know about fatty acids? Well, what do you know about fat? Fat is also a way to store energy. Fatty acids have a ton of energy. And where is that energy stored? In the covalent bonds. So fatty acids are used to make up phospholipids. Okay, phospholipids are what make up cells, and it turns out, to be really honest with you, the formation of phospholipids and the ability to segregate from water, the formation of an amphiphilic molecule that created a structure that allowed things to be separated from the extracellular space is what allowed life to occur. So when fatty acids were attached to this polar head group, and the polar head group has a particular structure to it that I don't ask you to know, but what I do want you to know is that that polar head group is polar. Polar because it wants to interact with water. It actually has a phosphate ion on it, and so this is highly hydrophilic. The fatty acid tails want nothing to do with water. So they want to minimize their interactions with water, and when you throw these phospholipids together in an aqueous environment, you actually spontaneously have the formation of either a phospholipid bilayer, shown here on the right. So the hydrophilic heads are interacting with water on the outside and the inside, and the hydrophobic tails, shown as the green lines, the fatty acids, they interact with one another. They're perfectly happy being together because of hydrophobic force. Hydrophobic force is nothing more than pushing water away. So in this case, you separate water from the hydrophobic nature of the fatty acid tails. Actually, I'm going to go back one slide and just point out the fatty acid tails in this image, right? One is straight, one is kinked. Does anybody know what kinked means? Kinked means that it has a double bond in it. And all phospholipids all phospholipids have one straight tail and one bent tail. And the reason for that is, and phospholipids that make up cells. Okay, you're going to see that there are actually some additional phospholipids that have more than 2 fatty acid tails. But in this case, this allows for the packaging of these guys to be not so tight. Okay, tightly packed would not allow for anything to get in or out of cells, right? That would be completely non-permeable. So the kink in these tails is really important for having permeability or semi-permeability of the cell membranes. And we'll talk more about that later. Okay, the third, right, we've talked about sugars, we talked about fatty acids, let's talk about nucleotides, okay? I'm saving the best for last. Nucleotides. Nucleotides are nitrogen-containing rings connected to a ribose or deoxyribose. Do you need to know the structures? Yes, but you only need to know the ribose or deoxyribose structure, and I'll show those to you in a minute. What do nucleotides do? Well, I hope you all know that they store energy, and they store energy in phosphate bonds. So not only do they store energy, as in ATP, or GTP, or CTP, or TTP, right? But nucleotides are really important for the storage and retrieval of biological information. DNA and RNA store information. DNA stores long-term information in your chromosomes. RNA stores short-term information in transcripts that are then used to generate proteins. Okay, let's look at the structure here. Okay, this is a pentose, right? If you start with the oxygen and you go to the first carbon, right, you just make a little pentose molecule. Carbon 1, carbon 2, carbon 3, carbon 4. The 5th carbon is off the top of the 4th carbon, right? And what positions are really important in this ribose? Okay, the second position determines whether you have a ribose or a deoxyribose. The image here has an oxygen on it, so that would be a ribose molecule. If it just had a hydrogen, it would be a deoxyribose molecule. That's what's going to determine whether you have a DNA or an RNA. Okay, the 3' carbon, all right, has a hydroxyl group on it. That hydroxyl group is really important because we make chains of nucleotides, right? Your chromosomes are made up of a chain of nucleotides, ACGT, 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 in some order. The linkage between one nucleotide and another is dependent upon having a hydroxyl at the 3' position. No hydroxyl, no bond is going to be made. So that's really important. The 5th carbon, right, the 5th carbon that's up on top, right, is going to be attached to phosphate groups, right? I told you that we can store energy here, and the energy is stored in phosphate bonds. The phosphate bonds are attached to that 5th carbon. So you need to be able to draw, this is just a ribose, right? It's a ribose because of the 2' hydroxyl, and right, if we actually add the phosphates on, and this is the image that I would like you to be able to draw, excluding the base, the adenine at the top, all right? But you should be able to draw the phosphate groups that are attached. So this would be, excuse me, if this has an adenine and it has 3 phosphates, this would be an ATP, right? Adenosine deoxyadenosine triphosphate. And why is it deoxy? Because at the 2 position, it only has a hydrogen. Right? What if it has an OH at the 2 position? Then it's just an adenosine triphosphate. Adenosine triphosphate is an RNA. Deoxyadenosine triphosphate is a DNA molecule. Okay? I know that some of you have never drawn these before. We're going to draw them. Hint, hint. You might want to study and learn these before Wednesday. Okay, that's what we're going to work on. You're going to need to know these molecules and how they're made. Now, what is the bond between each phosphate and oxygen called? Does anybody know? It's called a phosphoanhydride bond. Tons and tons and tons of energy are stored in phosphoanhydride bonds. Okay, so this is just another picture showing you the phosphates of nucleotides. The energy that's used, that's required to link one nucleotide to another, is stored in the phosphoanhydride bonds. If you don't have the phosphoanhydride bonds, right, and let's say you have AMP. What's AMP? Adenosine monophosphate. Okay, just has one phosphate. You can't attach that nucleotide to a growing chain of nucleotides. It doesn't have enough energy stored in it to make the covalent bond to the 3' hydroxyl. You need an ATP to make that bond. Okay, you do need to know what the bases are. You don't need to know how to draw the structures. I don't know how to draw the structures, all right? The nucleotide bases are A, T, right? A is adenine, right? It's called adenosine when it's bound to the ribose. A, T, thymine, thymidine. U is uracil, or I think it's uridine. Cytosine, and guanine, right? There's 5. Purines and pyrimidines, how do you remember which ones are which? All right, you always want a cut of pi. So C, U, and T are pyrimidines, all right? Pure as gold, A, what is it? A and G. God, that was hard. Pure as gold, okay? So purines are pure as gold, pyrimidines are, I want a big cut of pie. All right, with that, all right, what do you need to know? There's just that little reminder. You need to know how to link one nucleotide to another, and that results in a phosphodiester linkage. It's a condensation reaction. When you think of condensation, what do you think of? I think of water. So in this case, you're going to lose a water in this reaction. You can link these molecules together, and they're showing you here the nucleotide on the bottom. So you see an OH at the 3' position. What do you see at the 2' position? Nothing. Okay, if there's nothing, that means it's just an H. So is this a DNA or an RNA? Just an H, so that means deoxy, so these are DNAs. All right, and we're going to add at the 3' position, right? The 5' end of the chain is shown at the top, and we're adding to the 3' hydroxyl, and what you see is you end up with an OPOC. That is the phosphodiester bond. Phospho, right? P, di, 2, esters. Ester means you have a link to an oxygen. So there's 2 oxygen molecules. What's in yellow is the phosphodiester linkage, right? This is why we tell you over and over and over and over, you can only add nucleotides to the 3' position, right? You can't start adding nucleotides on the 5' end of a molecule of DNA or RNA. The enzyme that adds the base cannot recognize that 5' position. It can only recognize that 3' hydroxyl position. Think of how an enzyme works, right? An enzyme is a molecule that has the space for the reactants, so the 2 reactants sit in there. When they're sitting in there, it causes a conformational change, bringing those 2 reactants close enough together to change the chemistry. In this case, the enzyme that does this can only add at the 3' position. Okay, that, I believe, was our last slide. So you need to know this for Wednesday. I wish you the best of luck with that. We're going to have some fun on Wednesday, so I hope you get all this material. There will be a little quiz, so come prepared. Great.